Are we ready or what?
Yes, we're going.
Oh, okay.
It's okay, I can turn the video.
Okay. This is our DNA replication project by Emily Severe, Joshua Wynn, and Blair Bartholomew. DNA replication happens through a method called semi-conservative replication, where each of two partner strands in double helix act as a template for a new partner strand. Here you can see the beginning of original DNA and the product, the two new helixes. DNA strands are anti-parallel. Anti-parallel, pertaining to a molecular orientation in which a molecule or parts of a molecule have opposing directions. The first step of DNA replication starts at the origin of replication, where a DNA helicase enzyme unzips DNA, the DNA double helix, to separate template strands, where that is called the replication fork. Helicase, an enzyme that separates double-stranded DNA into single strands, allowing each strand to be copied. Origin of replication. Origin of replication, a particular sequence in a genome at which replication is initiated.
Replication fork, a point at which a DNA molecule is replicating. The fork forms by the unwinding of the parent molecule. The next step of DNA replication is when DNA polymerase III encodes and replicates the leading strand, forming from 5' to 3'. This is continuous replication because it is the leading strand. DNA polymerase III is the enzyme that catalyzes the formation of DNA strands from a DNA template. The leading strand is the daughter strand that is synthesized continuously. The next part of DNA replication is when the RNA primase lays down RNA primer on the lagging strand of the DNA. RNA primase is an enzyme that catalyzes the synthesis of a primer for DNA replication. The lagging strand is the daughter strand that is synthesized in discontinuous stretches.
In the third step, DNA polymerase III lays down new DNA on the lagging strand. In DNA replication, adenine pairs with thymine and cytosine pairs with guanine. As polymerase III lays down new DNA, polymerase I replaces the RNA primer with real DNA. These fragments of DNA are called Okazaki fragments. Finally, DNA ligase links the Okazaki fragments on the lagging strand. DNA ligase is an enzyme that unites broken DNA strands during replication and recombination. Okazaki fragments are newly formed DNA making up the lagging strand in DNA replication. After the process of DNA formation is complete, you have two new double helixes that are partially new and partially old. Thank you. Goodbye.